All right, we'll go ahead and get started with our third and final program for today's event. Again, thank you for being here. And thanks to our coaches and Kendra for facilitating our previous discussion. That was fantastic. I have the privilege of introducing Dr. Nicole Earhart, who is going to present some fascinating information about healthy aging. Dr. Nicole Ehrhart, she just let me know, which I think is fascinating, was the first woman to be granted a university chair in 2015. Did I get that right? Thank you. Which is remarkable considering that 2015 was not that long ago and was the first woman to be granted that role. So Dr. Nicole Ehrhart, excuse me, one second. Want to make sure I get this correct, is the director of the Center for Healthy Aging, and is here to present some fascinating information again about healthy aging and how science has decoded some aging on the cellular level. I'm butchering this, and that is why she is here to present on this. So everyone, please help me welcome Dr. Nicole Ehrhart.
Thanks for that really nice introduction. Let's see, is my lav mic working okay? Yeah, great. I just want to say what an incredible honor it is to be here among all of you and to celebrate this incredible day together with the amazing speakers that we've had so far. And I'm really honored and really want to thank the organizers for the opportunity to speak today, because I want to talk about some really exciting science that's been happening around how we age. And I don't know the ages of everybody in the room, but I will share my age. I will be 57 this year. Yes. And so I don't know how many birthday candles need to be on the cake before we start to feel old. I personally don't even feel grown up yet. I don't know how many people feel that way. Is it 50 candles? Is it 60? I don't know. And is 50 the new 30? I really hope so. That's what I'm going for. So I guess we want to know, really, is there a way to optimize our later years so that we aren't really burdened with the onset of age-related diseases, things like frailty or dementia, things that really kind of diminish our quality of life as we get older? And one of the things that I think we fail to recognize in our culture is that there are a lot of benefits to aging, right? I mean, we've got a more accumulated wisdom and experience. We have more emotional maturity and regulation. And we have a deeper contentment and satisfaction in the present than people that are younger. And definitely broader perspectives on current events. And one of the nice things, too, I think, about getting older, and I can say this because I'm a mom, is freedom from child-rearing responsibilities, right? I mean, I love my children, but boy, it's really fun sometimes to have the freedom to do what we want, when we want. And then freedom from some of the financial or resource building that we're really something that we have to build as we in the first part of our lives. So there are a lot of benefits. And of course, I love Betty White, may she rest in peace. But I thought this quote that she said was really poignant, especially for this particular talk, because she says the best thing about being in your 90s is you're spoiled rotten. Everybody spoils you like mad, and they treat you with such great respect because they know you're old. But little do they know you haven't changed, right? You haven't changed in your brain. You're just 90 every place else. And I think that's the thing, right? I mean, being 90 everywhere else isn't that much fun sometimes. And as we age, we begin to experience a decline in our fitness and our resilience, and we begin to accumulate some of these conditions that we associate with older age. And that can be a drag. And so for centuries, ever since ancient times, people have been really looking for this proverbial fountain of youth, a way to maybe reverse aging or slow aging so that we could avoid all that difficulty in our later years. And I love this painting. This painting was done in the 1500s in Germany, and it's a famous painting. Many of you probably are familiar with it. It's called The Fountain of Youth. If you just look at the painting and you see the people on the left-hand side are these women who are being carted down from the village in stretchers and, wheelbarrows, and they're asked to enter the Fountain of Youth, at which point they swim in the nude across the Fountain of Youth, and as they emerge all fresh as daisies without wrinkles, their gray hair is gone, then they're invited to go into the tent where they're dressed in exquisite finery and then go and have a feast and enjoy dancing, etc. And so while the Fountain of Youth has kind of eluded us, certainly, we have seen some things over this last 7 decades, that we should probably think pretty hard about. And one of those things is that, A, the world's population is growing. It's actually grown 5 times since the 1950s, or 5-fold. And since that time, the world population not only has tripled, but average human lifespan has increased from an average of 47 years in the 1950s, if you believe that, to now 76 years. That's enormous. That's an enormous increase in human lifespan. And so, but it also means that as the world population increases, a greater and greater proportion of the world is over the age of 70, and that's represented by the blue line there. So we see that increase, that exponential increase, and the pink line is people less than 70. So by the year of 2050, it's estimated that almost 1/4 of everybody living on this planet will be over the age of 65. It's kind of earth-shattering. And I would say that population aging is probably humanity's greatest triumph, because it represents a victory over so many things that used to limit human lifespan, like infectious disease and parasites. But it also represents a huge challenge, because there's a rapid decline now in people of working age, and therefore a larger number of people are depending on smaller and smaller pools of workers to provide health care, goods, services, etc. And just from a health perspective, one of the challenges that we are facing, and we are woefully unprepared for, is the fact that even today, 80% of people over the age of 65 have one of these conditions that we're seeing there on that infogram, that heart failure, chronic disease, kidney disease, diabetes, etc. And 68% of people over the age of 65 have 2 or more of these conditions. And if we just take one of the conditions that we associate with later life dementia, it's estimated that by the year 2050, the number of people living with dementia on the planet will triple. Our healthcare system in our society is woefully underprepared for that, and we need to think about this as a challenge that we need to address. So interestingly, when you think about it and you think about all this, we actually don't die of old age, right? Aging bodies are made of aging cells that are unable to fight and heal as they once did. And so as people get older, their cells simply just don't work as well, and they can't stave off disease as easily as they did when they were younger. So as a result, older people may die from injuries or diseases that younger people would have easily survived. So nothing really dies of old age. We die of conditions of old age. Now, our Western medicine, our culture, has really been focused on this disease-based approach, right? We wait until we have a diagnosable disease, and then we try to figure out how to treat it. But what if we took a different approach? What if aging is the one common risk factor among all chronic diseases in late life? What if we actually focused on that common thread, on aging itself, and started thinking about how we can change that? That would change everything, right? Because instead of thinking about, okay, let's cure cancer or let's cure heart disease, all really important things, if we were to do something about how we age on a cellular level, we can impact all of those diseases simultaneously. And so we've asked the question, can we change how we age? Can we target aging itself so that we can optimize our longevity? Can we live longer, but not only live longer, live healthier into our later years? So I'll give you a spoiler here. This Time magazine cover reported on a scientific study that looked at the human lifespan. And it found that were it not for the conditions that we accumulate as we age, if we could get rid of those conditions, our lifespan could be up to around 142 years. Okay, so we clearly have, as humans, an expiration date. I know it sounds crazy, but it's a lot further out than we might have thought. And it's the accumulation of these diseases that we get as we age that actually leads to our demise, not necessarily that we just have this expiration date. So if I were to ask people in the room to raise your hand today, if you want to live to be 142 years old, do we have any hands that would be up? A few, but not the majority, right? But if I were to say to you, what if you could wake up on your 142nd birthday, drive yourself to the tennis courts, play a couple of matches, come home, have dinner with the people you love, go to bed, and never wake up, and that was how your life ended at 142, a lot more of you, I think, would raise your hands. You see, it's not really about how long we live, it's about how healthy we live. And that's the concept of health span. It's the fact that we want to live the rest of our lives, our golden years, as healthy people who can continue to contribute. So health span is the number of years of life before a serious or debilitating illness begins to affect our quality of life. And what's happening in our modern world is that human lifespan and human health span have become uncoupled. So human lifespan is increasing, but human health span has remained static. So the field of healthy aging is really all about decoding why we age, and by decoding why, we can then modify the how. So if we know why we age, we can change how we age, with the goal being to increase health span first. The idea being to die young as late as possible, right? So that we spend fewer years of our late life burdened by age-related disease. So the first thing you need to do if you're going to be a person like me and you want to study aging is you have to sort of define aging. And you have to understand how we age on a cellular level, right? And so if we can think about this as maybe an opportunity to eliminate or decrease or delay the onset of age-related disease, the first thing we would do is define aging as sort of the lifelong accumulation of damage to our body, i.e. our cells, that happens as a result of the normal functions of our body. So it's that little micro damage that happens, the sunburns when we were 10 years old, and all of that accumulates over time that results in damage that becomes less and less reversible. And we know that living things can tolerate some damage, but if that damage is not reversed, it begins to result in failure of the system, and that is why we diagnose it as disease. The failure starts before the disease is actually diagnosable. Those micro damages are the things that are accumulating before we even know that that's happening. So we've always believed in years past that aging is this inevitable and universal process. And the reason we've thought that for so many years is the fact that we can observe aging on every single level. We observe it on the whole organism level. You can see a brain there on the left, you see a brain from a younger person versus a brain from a person of advanced age. You can see it on tissue level. So on the left, you see muscle tissue from a young person versus muscle tissue from an older person on the right. You can see it on a cellular level. You can even see it down to the molecules within our cells. The very DNA in our cells changes recognizably as we age. So we now have been asking the question, is aging modifiable at its most basic level, i.e. the cellular level? And I think if we can think about it as aging being the common element and the greatest common risk factor among all chronic diseases of later life, if accumulation of cellular damage is responsible for aging, could we possibly perform periodic maintenance on our cells to repair damage along the way? Can we fix that micro damage? And I like the example of a classic car. You can take a look at this car, and you can tell it's an old car. But if you were to lift up the hood and look under the hood, you would find that this car is running beautifully decades and decades and decades after its expected, quote, lifespan. Well, why is that? It's because someone has taken the time to painstakingly refurbish, repair, or replace those damaged parts. And now we have a car that looks old on the outside, but is running very well because it's been fixed. All the damage that it's accumulated over time has been fixed. So can we do that to our body? Can we fix that micro damage we've talked about along the way, thereby increasing health span? And now there actually is very convincing evidence that the answer to that question is yes, we can. And that's super exciting. So I want to walk you through the story that's been coming together over the last 10 to 15 years and how science is decoding this. So the story starts really by simply observing certain subsets of people. And I've got two examples here. These gentlemen are both 85. We have the guy on the top who looks like an athlete. In fact, I thought he was photoshopped, but he has his own Instagram following. It's like Fit Granddads or something. I don't know. He has a very big Instagram following. And then there's a gentleman on the bottom there that probably represents someone maybe more typical of someone in their later 80s. Not everybody. We all know very healthy 85-year-olds, right? But what's interesting is if you would think by observing 85 and plus age group that we would have this very even spectrum of people that sort of represent the ultra fit to maybe the ultra unfit, and there's this even number of people along that spectrum. But what we observe is actually something completely different. Actually, by the end of life, or in that 85 plus range, people tend to fall into two very divergent subsets. They're either super healthy, I mean, that guy's an extreme, okay? But super healthy out in the golf course, 5 days a week, or they fall into people that obviously have a lot more accumulated burden of age-related disease, like the gentleman on the bottom. Maybe they have mobility issues, maybe they have memory issues, et cetera. And what I've shared with you so far helps you to guess correctly that the person with the walker on the bottom has probably accumulated more of those micro damages we've talked about, right, over his lifetime. And so the burning question then is, can that be changed? Could we actually change our aging trajectory? None of us know what our aging trajectory is going to be like when we're young. We don't know that. So can we actually reverse a lifelong accumulation of microdamage? And so that's the question that we've been trying to answer. So to answer that question, we have to go back to the laboratory, right? And the traditional laboratory models of aging are things like yeast cells, worms, fruit flies, and eventually laboratory mice, okay? And we look to these traditional model systems to understand why some people age extremely well and other people do not. So as we know, nature and evolution is a very conservative process. For example, we see the same basic programming for basic life in just about every living thing, right? DNA makes RNA, RNA makes protein, proteins do the work of the cell. In every living thing, pretty much, okay? And just like the DNA story, in aging, what we do is we start with very simple organisms like a yeast cell, which has a lifespan of hours, and we start understanding what changes that programming of aging. And we work our way up to more and more complex animals. And what we see, remarkably, is that the same basic cellular events occur during aging in each of these animals, or each of these creatures. And these creatures have diverse lifespans, hours to years. The lifespan of a yeast is hours. The lifespan of a worm is weeks and, or days, and flies are weeks, and mice are maybe 1 or 2 years. So if we can look across all living organisms with very diverse lifespans, and we see the same basic programming playing out in each of them, what's happening is that that program is being executed in different rates, much more rapidly in the yeast versus the mouse. So this is huge because all of a sudden it gives us an idea that perhaps we can manipulate those dials. Maybe we can turn them up or turn them back down and slow things down. And so after years of many, many people's work and very eloquent science, we've now been able to drill down to understand that there are 9 basic mechanisms, just 9, that drive aging on a cellular basis. And you may recognize some of those if you're in the science field, things like telomere shortening and genomic instability and senescence of our cells. But those same basic processes cause aging in the yeast, as well as in the mouse, as well as in humans. And the accumulation of those 9 drivers of aging in our cells are what result in the age-related changes we recognize, our wrinkles, our age-related diseases, et cetera. So somehow if the basic aging program is the same, maybe the reasons why our 2 granddads we saw in the previous slide are so different is that the aging program is executing itself. It's the same program, it's just being played out at different rates in those 2 different people. So once we now understand what those universal aging programs are across all species, all of a sudden we now have an ability to potentially take our first steps to begin to slow that down or reverse it. Because those 9 things that drive aging are handles. They're knobs that we can turn up potentially or turn down with enough work. And I want to show you how this has actually happened, which is pretty exciting. So I love analogies, and so the best way to explain this is to think about our bodies like an orchestra. So when we're young, our cells and our tissues, like an orchestra, are perfectly coordinated, and they are playing together in a way that creates this highly complex but beautifully synchronized and composed musical piece. And then as we enter the later half of our life, a few of the instruments begin to go out of tune. Maybe it's the string section at first. And at first, the musical piece really isn't affected very much because the other instruments are so well coordinated, they carry the day. But then as we continue down that road of aging, more and more instruments, i.e. our cells, begin to hit the wrong notes, or they begin to not function quite as well. They go out of tune. And all of a sudden, the musical piece very slowly changes, and over time begins to resemble less and less like the original sheet music and become more and more chaotic until the original familiar melody really isn't recognizable. And in our analogy, this would be when we begin to start experiencing disease or dysfunction, and eventually that music stops because it becomes so chaotic it's not even music anymore. Okay? So let's go back now to our two granddads. We now know something about the cellular processes that are going on in these two people. They're described by the 9 hallmarks we've talked about. Those are the progressive changes that happen on a cellular basis that lead us to higher risk of age-related dysfunction or disease. And in this example, we know that if you were to measure the cellular hallmarks or drivers of aging in the gentleman on the right, his orchestra is still hanging together pretty well. But versus the man on the left, where we're seeing a lot more chaos, a lot more of those drivers of aging being accumulated on his whole body's basis within his cell. So what we know right now is that somewhere between the age of birth and 85 years of age, these two people began to diverge. They began to go down 2 different pathways, one a very successful aging pathway and one a less successful aging pathway. But as time went on, they became less and less alike and more further apart. And now we see this snapshot at age 85, which really shows 2 very different end results. So this observation leads to some really important questions. Can we minimize that divergence? And if we think the answer is yes, we could bring those 2 people less farther apart, the question then becomes, when did they begin to diverge? Was this happening when they were born? Like this was just their genetic deck of cards that they got, that one was going to go down this aging pathway that was less healthy versus others? Or are there ways in which they began to diverge further and further in the span of their lifespan, at age 30, at age 50? And the premise is that if the accumulation of those cellular drivers is responsible for the aging program playing itself out differently or more rapidly in the gentleman with the walker, could we actually have a different outcome if we blocked those drivers of aging, some of them? Okay, so if we could block those, could we move the gentleman with the walker more to be like the highly successful ager? And of course, to know that, we'd also need to know when would be the best time to begin to treat the difference that was occurring between these 2 people. Can it be effective only if we start at the very beginning when they begin to diverge? Or would we be able to actually reverse some of those changes even at age 85? And we have no way of knowing that, right, in humans, because we don't know when they diverge. So of course we go back to our trusty laboratory models of aging, the yeast, the worms, the flies, and the mice, and we start thinking, okay, in the simple organisms, can we modify those drivers, and does that make a difference? And if blocking drivers of aging works in yeast and worms and flies, and we're really lucky and we figure it out, then we move to the mouse. And this actually has happened. We take an old mouse, we give that mouse something that blocks one or more of the drivers of aging, and we hopefully reverse the cellular aging program. And in fact, that's exactly what you're seeing here is the results of those experiments. So what you see on the right-hand side are 2 mice. The mouse on the left, they're both old mice. The mouse on the left is showing typical signs of an old mouse. You can see the hunched appearance of the mouse. You can see the muscle wasting and frailty, the loss of hair. The mouse on the right is exactly the same age. It is a litter mate born on the same day of the mouse on the left. The difference is that the mouse on the right has been given a blocker of one of the drivers of aging. In this case, for those of you who might want to know, it was rapamycin. Okay, I don't know if you've heard of that. And look at this mouse. This mouse looks nothing like the mouse on the left. So it's very similar to the 2 granddads we talked about. And in fact, not only on a whole organism does this animal look different, at a cellular basis and on an organ basis, this animal is younger, okay? So that's the thing, is biologic age is not coupled to chronological age. We thought it was, but it's not. We can undo that, okay? So, of course, Aging is actually modifiable. We've just shown that by looking in the mouse and the yeast and the worms, et cetera. And by reversing or significantly slowing cellular aging programs, we can now delay the consequences of aging on a whole organism level in a mouse, okay? So this is huge news. And actually, when this was first published, the media picked up this story. It was talk show material. Scientists have found a pill that reverses the signs of aging. People were high-fiving in the lab. I mean, it was really, really exciting. And 8 years has gone by since that was published. So what's going on? Why haven't we heard about these incredible treatments that are supposed to be helping us age in a more healthy way? And if you're asking those questions, those are really good questions. Well, unfortunately, this story has been repeated in science and medicine many, many times. And what happens a lot of times is that because things are successful in mice, they don't necessarily translate that well in people, right? I mean, mice are nothing like people. They live their entire lives in a laboratory setting. They eat the same diet all day long. They never go to McDonald's and get a Shamrock Shake. I like Shamrock Shakes. That's the only times I ever go to McDonald's. But you know, people are nothing like that, right? They're highly divergent. They're epigenetically and genetically completely different from one another. And our health habits, our socioeconomic status, our access to health care, clean food, clean water, all of those things influence how we age. And we can't mimic that in a laboratory setting, right? So all this discovery is piling up on one side of this big chasm, and we call this chasm the translational gap. It's the failure of discovery to make it into meaningful health changes into people. And we've been burdened by this for many years in many fields of science, not just aging, but certainly aging. So we're not able very well to translate those findings from traditional laboratory models into people. And what we need is a bridge. We need a bridge across the translation gap. And that bridge has been largely unrecognized until quite recently. So to review, let's look at our two granddads again. We now know that something about the man on the left has caused his cells in particular, to age more slowly. We know that at age 85, the man on the right has many more accumulated drivers of aging in his cells. And we know that conceivably, because we've been able to do it in a complex mammal like a mouse, we potentially will be able to reverse aging. But we need something that's going to model or mimic human aging in a better way, okay? And if we can do that, then we can move those discoveries across the bridge and change the man with the walker, hopefully to end up a lot more like the very fit granddad. But we're missing two essential pieces of information. The missing puzzle pieces to this story is that A, we don't have a model that mimics human aging well. Those traditional models don't work that well. And we need to know an answer to a very crucial question, which is, when during our life do we begin to go down the wrong path, the less healthy aging path? When does that happen? We've already said that by age 85, people tend to fall into two very distinct groups. So did that divergence start at birth? Did it start at age 30, at age 50, or age 70? And that information is crucial because we need to know if intervening with these blockers of the drivers of aging that we've been talking about needs to happen super early on, or can it happen late in life and still have the same effect? The problem is in humans, we just don't have a good way of knowing that. I mean, the only thing we could do maybe to answer that question is to take a large group of people, and from the time they're born to the time they die, take cells from them every single year, look at that group of people, see who's the healthy ager and who's the less healthy ager, and then look backwards and see when did their cells begin to diverge from one another. That's the only way that could ever be done. It's never been done. I guarantee it won't be done because it's too expensive, and it would take 100 years to get the results, right? That's just not how science tends to move forward. But there is a better way. So I'm going to show you two more examples, and these are two dogs. Both are 9 years of age. And on the left, you have a Jack Russell Terrier. This dog is an athlete, the national flyball champion, actually. And for those of you who don't know dogs very well, the Jack Russell Terrier is a little dog. They're usually about 12 pounds or so. And then on the right-hand side, we see this Great Dane, giant dog. And this dog is, unlike the dog on the left, is showing signs of advanced age, even though she is the same age as the Jack Russell Terrier. I don't know if you can recognize her hunched stance, similar to the mouse that we saw on the other slide. muscle wasting, and by the way, she happens to have heart disease and kidney disease. So at age 9, this Great Dane is a geriatric dog. And at age 9, this Jack Russell Terrier is in this prime of life. And for those of you who know dogs, of course, you know that canines have predictably divergent lifespans based on body size. So dogs that are really large live 2 times less than dogs that are the toy breeds, 2 to 3 times less. And this is really kind of an interesting phenomenon because for most of the animal kingdom, big animals live longer. Think of a hamster versus an elephant. That in most of the 99% of the animal kingdom, larger body mass is associated with a longer lifespans. But the dog is absolutely unique. The dog is unique in that larger body size is associated with shorter lifespans. And this is because the cellular aging programs are accelerated in large and giant breed dogs relative to small breed dogs. And dogs age naturally, right, alongside of us, right? Unlike mice, they get all the same diseases of aging, but just naturally. They get arthritis, they get cataracts, they get heart disease, they get cognitive impairment, just like people do. And also, unlike laboratory mice, they sleep with us. They share our food and water. They're exposed to the same environmental stresses and lifestyle choices like active lifestyle versus a sedentary lifestyle, urban versus rural environments. And just like humans, they are genetically and epigenetically very different from one another, similar to humans. Plus, they have access to an advanced healthcare system into old age, just like people. So laboratory animals don't have that. And they age predictably either rapidly or slowly based on their body size. In fact, if you look at what has been published about why large breed dogs have such a short lifespan and reference that back to those drivers of aging, what you find is that the giant breed dogs have all those accumulated drivers of aging, just like the granddad with the walker, okay, relative to small breed dogs in the same age group. So now we have an example that's unique in all the animal kingdom of a creature who lives with us, is exposed to our lifestyles and environments, who develops diseases naturally like we do. But unlike people, where we have no way of knowing when they began to diverge in their aging pathway, we know exactly when that happens in the dog. When the Great Dane and the Jack Russell Terrier are puppies, they age at exactly the same rate in perfect parallel. But as soon as they become skeletally mature, which is at about age 1, all of a sudden the Great Dane ages in rapid motion relative to the Jack Russell Terrier. And no other animal has this unique, predictable divergence. So this is very, very powerful because all of a sudden it gives us an opportunity to be able to study interventions at a known point in divergence. We can study them very early. We know when they start to diverge, right, at skeletal maturity or 1 year of age. We can study that a year later, 5 years later, or at late age. In a time frame that isn't 100 years long, it's 10 years long maybe. So this is a huge, huge platform for aging research. But the best news of all is that this is not just for human health. This is completely a two-way street. How many people in the room are dog lovers? Okay. How many of you then would not leap at the chance to see your canine family member age in a better way for longer and healthier, right? And so we're not only do we have a unique model, the companion dog, right, with which to study aging, this can be done certainly to benefit not only humans, but also dogs. And because of our special relationship with dogs, that they live with us and they eat and sleep and breathe with us, combined with the fact that their natural aging process occurs just like it does in people, and that uniquely in all of the animal kingdom, we can only in the dog predict when the aging programs begin to speed up. Can't do it anywhere else. That is huge. So it turns out that this missing bridge across the translational gap has been growing old right beside us all this time. And we've really failed to recognize the power of that until very recently. So CSU has some very unique strengths that are unlike any other research facility or any other, aging research institute. MIT has an aging research institute. Harvard has an aging research institute. None of those aging research institutes have a clinical trials program designed for dogs. And it started in the cancer world, and I worked there for most of my career. But basically, it is a research program where people can volunteer their pets to participate in longevity therapeutic studies. Completely voluntary. It's not like we're experimenting on pets. This is something that if owners would like to try this, and all the same clinical and ethical tripwires built into those clinical trials so that we can make sure that those animals get perfectly good health care, perfect standard of care, that if there's any side effects that we treat all that, just the same that would happen in someone signing up for a human clinical trial. That is what we do extremely well at CSU, and we've been doing it for years, and we do it better than anybody. And so we have that opportunity, and that is something we are building on because we're creating this translational aging research pipeline. And so basically the concept is we can take old mice and make them healthy and feel young again. We can take old dogs and make them healthy and feel young again. And hopefully what we learn in those beautiful creatures that are our best friends, we then take those things and make older people feel young again. But remember, this is still a two-way street. So what we dial in the dog benefits people, and what we dial in people benefits dogs. And we have the ability to do those studies in parallel, in people and in dogs, at the same time. Nobody else can do it that way. CSU is the only place. So we're developing this comparative canine aging intervention program with 3 core activities. The first being what we've talked about, intervention trials and companion dogs to help dogs and humans live longer, healthier lives. Global education to educate medicine and researchers about the benefits of this kind of one medicine approach to the grand challenge of aging that we are facing. It's right on our doorstep. And then we're also developing a biobank. This will be the world's first 5-year longitudinal paired human canine biobank. So dogs and people living in the same home and taking different samples from them on a yearly basis to look at biomarker discovery. Are dogs actually sentinels of our aging? If what we see is happening in the dog, does that actually predict what will happen in us? Or vice versa, what's happening in us, will it predict what's happening in dogs? Because we're living in the same environments, right? Those same determinants of aging occur in both of us. So I want to end this with a personal story. Some of you may know I'm actually a surgeon by training. That's been how I spent most of my career. And I've had the very great blessing of having many really amazing mentors in my life. And because of that, I've really had an opportunity to think outside the box about how human and veterinary medicine are so linked. And they're really the different sides of that same coin. Cancer is cancer, whether you have 4 legs or 2 legs. Same with aging, whether you have 4 legs or 2. We age the same, just as I showed you. So I often say that because of my mentors, I've had the opportunity to stand on the shoulders of giants, and I think that's very true. And the man on the right is one of those mentors, so I want to share with you a little bit about him. His name is Don Piermati. He was a faculty member here for probably 35 years or so, and was one of my mentors as a young surgeon in my residency. I thought this man walked on water. He was such a kind and patient guy, and he was in some ways a father figure to me. I really adored him. And he retired during my surgical residency here at CSU, but he always took an interest in my career. So we kept in touch, and he and his wife, Marcia, and I became friends over the years. The picture that you see on the right was taken when he received a Lifetime Achievement Award at the end of his career into his retirement. And that picture was taken about 17 years ago. So this is a picture of Don and I that was taken about 5 years ago. And it was taken only about 48 hours before he passed away. He passed away from cancer. And he is at home. He's comfortable. What you don't see in the picture is his 2 little rescue dogs that are curled up beside his legs on that hospital bed there. And he was in hospice, and I knew he only had a short time left, and he knew that. So I came to visit him, and we spent quite a bit of time just reminiscing about old times, about the funny stories, the funny clients, the poignant stories, the sad stories. And at the end of this visit, he sort of got very quiet for a minute, and then he said, I wish I could still be in the OR. I really wish I could still be doing science and still making a mark. And he made quite a mark in our field. Seems too soon to be done, is what he said. And I agree. I think it was too soon. I wish I could have given him more time to continue to do what he loved. He loved training residents. He loved advancing science. He was a pilot. He built his own planes. I mean, this man was an amazing person. And the only thing I could really say at that point was this reassurance that his legacy will always live on in me and everybody he taught. And that what he leaves behind is not what's engraved in a monument. It's what is woven into the life of others. And so even just about 5 years later, I think the ending can be different. And very soon, I think we're going to be able to give people more healthy time so they can continue to weave that wisdom and experience and the actual love and care that worldly wisdom gives us into the lives that they love, to continue to do what they love, and so that the world can continue to benefit from great minds like him. And I truly believe that the creatures that we share this planet with, the ones especially that share our homes, hold the key to our health and our longevity. I really believe that. And by partnering together and reaching across scientific disciplines, that will be the path forward, and that's the way we're going to change the ending. So thank you. I think we have time for questions, yes?
Yeah. I did forget to mention in my introduction that we will have some time for questions, so I'm happy to pass the mic, or you may speak loudly, whatever you're more comfortable with.
Obviously, the field of genetics is very far from being deep into what they study. I have juvenile diabetes, so since the age of 3, my pancreas hasn't worked. I see that you're working a lot with cancer patients, and obviously dogs helped us develop insulin. Do you also see this playing into other fields, especially around juvenile diabetes? I mean, I also believe a cure is in my lifetime, and this could be the way there.
Right. Yeah, I mean, many of the diseases that humans suffer from, especially chronic diseases like diabetes, are mimicked very, very similarly in dogs. And of course, now that preventative disease like vaccination, even in our companion animals, helps them to live into their old age, we have the privilege of actually having this population of old dogs that we can study. And you talked about genetics, and I think it's a really important point to bring up that genetics is, again, I love analogies, right? So genetics is like the deck of cards that we're born with. Everybody's are different, okay? But we have this deck. And epigenetics, which is the level above genetics, how those genes are expressed, is kind of the hand that we get played. So if you have a deck full of jokers, let's just say, which maybe the propensity to develop a disease like juvenile diabetes, the chances are that those might come up in a hand are much higher than someone who doesn't have that. But the good news is that genetics doesn't tell the whole story. The epigenetics, how the hand is played, can be modified. And we've shown that exactly with these experiments, that the cellular drivers of aging are really epigenetic changes, okay, for the most part. And we can actually turn the dials on those in ways we never could do that before without the understanding that we now have of the cellular biology of aging. So just like diabetes, and diabetes is one, and there are many other chronic diseases, by understanding those basic mechanisms and understanding what we can actually, it's not just about the genes, because we can't change that, but we can change how the hand is played. So I agree with you that I think we've got a lot of promising things in the future that will really help you as well as others with other chronic diseases. So thanks for that question. Yeah.
You were talking about the point of divergence. If you, like in the dog, it's about a year. If you go past that point of divergence without any trying to intervene in the aging, are you just past the point? Like, is there still a point in which you can reverse it, or are you just down that road?
Yeah. So the question was, for those who couldn't hear, is we talked about the point of divergence in the dog, which we know is at about 1 year of age or skeletal maturity. And the question was, if we start to intervene past that time of divergence, is it too late? Did I get that right? Yeah, and what's actually really promising about the field of healthy aging is it's turning out that there's actually a benefit to this very late in life. So even far beyond that point of divergence, we can begin to reverse those changes. And so again, it's harder to do in an 85-year-old that has that much more accumulation of that microdamage, but it still helps them. And so it's been shown in mice to be able, like even mice that are very much at the end stage of life can actually, they can reverse some of those age-related changes. So that's very promising. Again, still not there in humans yet, but we need to figure that out through, I think, the companion dog, which is our bridge. So great question. Yes?
Thank you for being here, doctor, and updating us. I didn't realize you had these studies going on. It's wonderful. So I participated in 2 aging studies with Dr. Deal here on campus, and one of the things that he brought to light was BDNF, brain-derived neurotropic factor, that could influence our dementia or kind of stave it off. And so I wondered, are you familiar with that, and are we anywhere along the path of maybe synthesizing that? And then the second question is, reference the genomes. Are we using CRISPRs for animals as well?
Okay, so the question was, the first question was relating to some studies that you've participated in with Dr. Monfred Deal, who has a study going on in perceptions of aging and how we believe our aging process is inevitable, and when we actually are told differently, it changes how we age. And some of what you were talking about is the idea of certain chemicals or neurotransmitters. You brought up, what was it? Brain-derived growth factor, or what was it?
Brain-derived neurotropic factor.
Right, brain-derived neurotropic factor. And the question was about, are we doing anything similar in dogs? And the answer to that question is yes, we are. One of the things that we're doing, and this is via some work that's being funded by the Vice President of Research here, is looking at certain packets of information that float around in our bloodstream that actually drive cellular inflammation, specifically in the brain. And we can actually find those markers in dogs, and we find them in people. And when we do that, we can actually either target those markers or actually remove some of those little packets of information and actually influence how animals age. So that's one. So it's not exactly synthesizing the same thing, but it's creating that same effect, or getting at the root of what drives the brain inflammation that then eventually causes dementias, et cetera. And then the second question was?
Are we using the CRISPR?
Oh, CRISPR. Yeah, so CRISPR gene editing, probably you've heard of this. The Nobel Prize was given recently for CRISPR technology, where we actually can edit genes. So we talked about how we can't really change our genetic deck of cards. Well, there is evidence that we can actually gene edit certain things out of our genome. It's still, again, very early research, but there's hope anyway that things like juvenile diabetes or other things that you are born with really could be edited out of your gene, or even just in a single organ, so in the pancreas as an example. So thanks for that question. That was very, very helpful. Yes?
I'm not a scientist, so this may sound like a very basic question, but I'm curious that dogs that are larger will age and faster and die earlier than smaller dogs. Is that because of the way we developed them? Because we created all the dogs that are out there?
Yeah, it's a great question, so I appreciate you asking it. So the question was, for the audience, if you couldn't hear, is the reason why giant breed and large breed dogs, why they age more quickly, a man-made phenomenon? Did we just create that by creating our breeds? And the answer is nobody really knows. I mean, certainly we have created breeds, right? But what in that creation caused cellular aging programs to accelerate? That has not been answered yet. So probably the answer is yes. We may have created that inadvertently, and maybe that's going to be, in the end, our salvation in some respects, and maybe theirs too. You know, who wouldn't want their Great Dane to live to 17 and be perfectly healthy, right? So it may be kind of the happy accident that we never wanted but ends up helping us type of thing. Yes?
How can we stay informed of this research?
Oh, thank you. She asked how we can stay informed. The best way is to go, first of all, you can go to our website, Healthy Aging, at coloradostate.edu, at colostate.edu. Website's always being updated. Sign up for our newsletters. We'll keep you apprised of all the clinical trials that we're doing, both for people and for dogs. Fundraising opportunities, ways to get involved just as volunteers, et cetera. So please, if you get a chance to take a look at that, and I have some business cards I can hand out for those who are interested. Check out our website. That's probably our best way, and then sign up for our newsletter. And I really appreciate that. Did you have another question too? What was that? That was it. Oh, supporting, yes. And so the canine intervention trial program that we're developing, we are fundraising for currently. So that's always an opportunity. You know, if you can donate time, just spreading the word, financial support, any of those things, or participate in trials. All of those things are ways to contribute and support what we're doing. So thanks for that question. You had a question back there.
So I have a question from my daughter who couldn't be here today. If you were 22 years old and going to go get a PhD in biochemistry and molecular biology, knowing what you know now, what would you focus on?
Whew. Wow. That's a 2-bottle-of-wine conversation, I think.
White or red?
Wait, red. Well, I like both. No, I think I really appreciate that, and the fact that she's asking that question is really amazing. I do think that the field of aging research, molecular biology in particular in aging, is exploding right now, and that aging-related everything is going to be a huge niche market. Aging-related engineering, aging-related therapeutics, aging-related everything, because of what we're facing. I mean, look at our world. These are the people that are inhabiting our world, and we're all over the age. We're going to be all over the age of 65 in 20 years or more. So she'll be right in position to be at the cutting edge, I think. So I mean, obviously I'm biased, right? But I certainly would love to have a bottle of wine and perhaps dissect that a little bit more. So that's a great question. Thank you. Other questions? I haven't seen any from over here yet. I don't want to leave anybody out. We'll have, one more.
Sure. Only because I hope it's fun. What's your makeup? What's your skincare routine? Very much so want to meet your dermatologist.
Oh, well, really the trick is you stand around in kind of shadowy places so people don't see the wrinkles, and you drink a lot of red wine. Yeah, thanks. No, that's really sweet. And I will say, if really what is at the root of the question is what can you do today, what everybody can do today to age in a more healthy way, you need to move every day. You need to be active. Motion is life. Get good sleep. Mindfulness and reducing stress, all of those things are things we absolutely have control over. Quitting smoking, that type of thing, are things we actually have control over that can affect how we age. But we're going to go deeper than that. We're not stopping with what you can do just today. We're going to continue to work at this till we get to the real sweet spot, and perhaps maybe the fountain of youth. Who knows? So, thank you, everybody.
I just have a couple of closing words. Thank you to Dr. Nicole Ehrhart for that incredible presentation. I definitely learned a lot, more than I thought I would. And then our thanks to our other participants as well, the coaches, Kendra, Kim Tobin, President Joyce McConnell. I know I'm forgetting somebody, but also thanks to all of you for joining us today. We really appreciate your time and commitment to joining us for programming like this. And just some final words about women in philanthropy. You know, we are really committed to engaging our community with events like these that shine a light on the incredible work happening at CSU, like the work of Dr. Nicole Ehrhart. And we encourage you to explore ways that you can support the people and programs that you learn more about today. And thanks for gathering with us, and we hope to see you again in the future.